Josh Winscott, and I'm here to present my research experiment over jet alert's effects on glycogen phosphorylase B's reaction kinetics. Before I get into the nitty-gritty of this experiment, let me give you a visual. It's 9 a.m., you just had breakfast. Lots of carbohydrates are going through your system. Your body has high insulin and high ATP. Go through your day, classes, studying, the usual. Maybe a workout. It's about 5:15. You're hungry, and you notice your stomach grumbling. Well, your body's filled with high glucagon and high levels of AMP, key aspects of low energy crisis inside your body. So what enzyme is working? And that enzyme is glycogen phosphorylase, specifically GPA, or active form of this enzyme. What is it doing, you ask? It's breaking down glycogen to gluc-1P, so it can further break down and metabolize into glucose and potentially ATP. So it's about 5:30. You realize you don't really want to eat anything, but kind of need some energy. So you pop a little caffeine tablet known as Jet Alert Double Strength. It's chock full with caffeine. How does it affect glycogen phosphorylase B? I'll let you know. So to get to our results, we had to first set up various assays, and specifically they are Michaelis-Menton kinetic assays. And what that is basically we keep the enzyme concentration the same and increase our substrate concentration to find the reaction velocity in Km. So this setup is a little different than what we would actually see in our body. So for our body to break down glycogen, it needs gluc-1P to be pulled away from the reaction zone, and therefore glycogen phosphorylase would act and keep hydrolysizing more and more glycogen into G1P. However, we don't have long strands of glycogen, and it's very hard for us to replicate this product loss and product pull-through, if you will. So we ran the reaction in the reverse order. So now AMP is the activator, G1P is the substrate, and the product is inorganic phosphate. So we made a standard inorganic phosphate curve to get an idea of our absorbance patterns and how they react with our stopping reagent. So let me describe this workflow to make things more clear. The assay starts with an addition of 50 microliters of GPB. We wait 30 seconds, and then we have another reaction addition here in the second cuvette. And it goes on more and more, and then there's more additions every 30 seconds until all the cuvettes have our enzyme. This reaction runs its course. And then we add STP. STP changes the color of the solution in response to how much phosphate is within the reaction vessel. So there, we wait 10 minutes, we let this color change occur, and then we make absorbance pattern readings. And from there, after those readings, we create our calculations, and then we create our Michaelis-Menton plots. All right, so here are my results for the kinetic assays. Up at the top, we have our raw data, and then in the middle, that is my corrected data, and with the red box surrounding it, that is the experimental jet alert effect on GPB with 4 micromolars of AMP. So for starters, we put 4 micromolars of AMP in our experimental, in the middle, our experimental jet alert assays, and then our negative feedback inhibitor assays. And that is consisting of glucose 6P. Glucose 6P is a downstream inhibitor of glycogen phosphorylase. And when it is in around this enzyme, it slows down the reaction velocity. It increases the Km, which means it's increasing the binding affinity to substrate to stop it from producing product. So all you see here is a clear-cut negative inhibitor of glycogen phosphorylase B. AMP is, our results were definitely a little spotty, and that's a lot attributed to this graph and this graph here. I also had a mistake on the absorbance readings, so that had a lot to do with more spottiness in our results. But we had good results for the 4 millimolar AMP assays of AMP, and we use that as a way to depict our data. So in 4 millimolar AMP, we see a decrease in the reaction velocity. And that just goes to show that jet alerts actually inhibit glycogen phosphorylase B. And it's seen right here when you have only 0.5 micromolars of AMP. You again see this inhibition and an increase in Km, which again means that substrate is binding very strongly to the enzyme and it's not able to produce product. All right, so just kind of reiterate the results of the experiment. Jet alert definitely inhibits GPB, and that's just because it resembles the assays of the known feedback inhibitor G6P. And if you think about it, it all kind of makes sense. Why would glycogen phosphorylase B, when it's getting sensations of wakefulness from caffeine, want to break down glycogen to create glucose and produce more ATP? Doesn't make sense because we're having in our body the sensations of high energy. And so that all kind of leads back into jet alert inhibiting GPB. And it makes sense in that regard. So in the future, I do want to see how jet alert impacts other metabolic enzymes. I would love to see these results repeated and reran in order to improve our accuracy. And then I want to see what other potential GPB inhibitors and activators compare to these results. Anyway, thank you to Dr. de Guzman for being my mentor and my wonderful lab partners right here. And Jet Alert, feel free to sponsor me. All right, thank you.